platform here. If you can hear, please type 1 at the question tabs there before I can start. Okay, can you see a question tab? So if you can hear me or listen to me, please type 1. Okay. Very good, very good. Yeah. Thank you. Okay, since everybody can hear me well, so I'm now. Okay. During the if you have any question, any issue that you would like to voice out, so we encourage you to type your question at this questions tab, because this is to avoid the interrupt during to present the. So I think this is easy. I saw everybody can type the very well. So okay, before start, let me. I'm Jenny Liu, okay, customer relationship manager from Epical Scientific. Okay, first base is our manufacturer branding. So I believe all of you who attend this get our EDM, or you might get from Epical, our distributor. All the past working very well with Genetical and build up a very good foundation to provide the lab service to our customers in Indonesia. Okay, so we actually would like to show our appreciation to our customer by giving provides useful for you all. One of the basic fundamental about the Sanger DNA sequencing might able to help you all in your research lab. So today, the title of my presentation is how to maximize your success of Sanger DNA sequencing with first base. Okay, so let's move on. So today, I'm going to cover some of the content of many of us know first base, but in order for you all understand better how first base as a manufacturer to the good service or the promise service or quality services to our customer, we would like to let customer understand better how is our workflow and how is what is the step or what's the guideline that actually we can give you, so that in the future when you send in the DNA sequencing sample, actually you can control it better and get the maximum, get a good result in your research. Okay, so let's move on. Okay, overview about first establish. Sorry. Okay. First base actually established in 2002. Okay, if you calculate the number of the years, actually, we have been in this market for about already. Okay, we have been leading the life science product manufacturing in the Pacific region. So some of the customers on our international side included Thailand, Indonesia, Philippines, and some of the other countries as well. So our mission is we would want to focus on provide the satisfaction to our customer. Okay, so side of as a service provider, become a leader in the market of life science, but we more concerned about on our customer demand. Okay, that's what specialty makes us different from other service provider. We not only provide the service, but we ensure the service actually is up to standard, and we able to help our customer whenever they need the help from us. Therefore, we also have a lot of team, okay, cascade team, and we also have equipped with our logistic team, such as dispatch. Okay, we also have a manufacturer facility in. So these two facility is the main facility lah, under first phase. Okay. Okay. 2012, we also but now other company called IDT lah. Okay, so top biotech company in our home market, and we also build a very good with our other distributor from all over the country as well. Okay, what is the main product and service that we first provide so far? Okay, the slide you can see, actually we not only provide Sanger sequencing, but we do a lot of service that related to a molecular based service, such as next gene sequencing, which is I think a lot of actually getting keen to use or to try to approach this service now. So we also have analysis service, molecular biology service, which means that we're able to help customer to perform a DNA service or subcloning, cloning service as well. Okay. So have our first base brand biochemical buffer and reagent. If you have never used our first base reagent, we highly recommend that you can understand more or get more information from our distributor under Genetic Hub. Okay. Okay. There's other service which is also we are covered, like metabolomics, peptide synthesis, antibody production, and proteomics services. Okay, whenever you need any support or any inquiry that you want, just go to our distributor. I believe Genetica will be willing to help you all. Okay. Okay, let's come to the topic, the main topic of today, in Sanger DNA sequencing service. Okay, so I think majority of from molecular biology science. Okay, so Sanger sequencing and PCR definitely is the term that not very odd to you all. Okay, PCR and Sanger, as you know, actually is the meaning of definition is different, but it looks quite similar. Okay, but let's do a revision. Okay, a very general revision only. Okay, wait, let me open back my. Okay, sorry about that. Okay, so if you can see, there actually is a difference between PCR and Sanger sequencing. Okay, what is PCR? Okay, PCR in general, we can say it's a technique that used to duplicate the DNA artificially, right? Okay, so in order for customer to send us a sample, I think with the DNA that you extract from the sample definitely is not enough for better to proceed further like sequencing. Okay, that's why what to do is they will proceed with the perform a PCR before you all send to us. Okay. Actually, PCR also fundamental technique that by you all. I think this is not is a very familiar technique. So this technique actually involved in two, which is forward and reverse, basically, right? So without these two primer, I think you're unable to amplify any the PCR product. Okay, and after that, definitely you need to the NTP as well. So that when you put it into the PCR machine, it can duplicate the PCR product. Okay, then what about Sanger sequencing? Okay, actually Sanger sequencing is come from a process whereby the sequence of the base actually is for determined for a research or medical use. Okay, so this technique or technology actually is also so-called chain termination method. Termination method. In this method, the target DNA is copied many times. Okay, same like this, but it's a linear rate reaction. Means that it only duplicate in one way only. That's why it in only one primer. Okay, you see here one primer of two primer if we refer back to PCR. So Sanger sequencing actually the technique is it try to copy many times to in a different length. So by using the fluorescent chain terminator nucleotide to mark the end of the fragment and allow the sequence. Okay, if you can see from here, the of the PCR and the structure of the Sanger sequencing is a bit different. Okay, so if PCR, you can see actually the structure included the OH, OH group. So in this group, the DNTP actually can form it continuously. Okay, but if Sanger sequencing, the structure of the chemical itself actually hydrogen group. Okay, from here is involved in the DNTP. So this mechanism actually is important for us to terminate the string to get the end of the fragment. So if you see here, okay, this is where you can see, okay, the sequence fragment can be determined accordingly by using the method of Sanger sequence. Okay, nucleotide such as ACGT will be included in the reaction as well, and polymer plays a very important roles. Okay, to replicate the strength of the sequence. Okay, so from this diagram you can see actually nowadays the sequencer we already most of us upgrade to the capillary model. This certainly is happened in first place as well. So from the capillary sequencer, actually it involves some fluorescent detection by giving an imaging. So you can see the here. So whenever the fragment ends, the nucleotide will capture chromatograph, okay, which is you can see in the part B here, okay. So this is the fundamental of the Sanger sequencing, okay. So after that, let's come to the workflow of Sanger DNA sequencing. In whatever service provider, the workflow of the Sanger sequencing actually is the same. Okay, so the first step is involved about the template purification. So this step basically will need to perform by our customer or researcher that send us. So in this step, we must always make sure that the sample that we amplified must be purified before you send to Sanger sequencing. Okay. From this PCR reaction need to undergo a purification. So how you are going to do that, you can either use the purification or you can use the cleanup. Okay, so this is the main purpose is to ensure the salt, the excess primer, and other contaminant won't spoil the sequencer when it load into the okay. So in this part, please remember that only purified PCR product plasmid can be used in the Sanger sequencing. Okay. So after you have purified your sample, the second step will be the cycle sequencing. Okay. What actually means that about cycle sequencing? So in cycle sequencing, it is involved about reaction that involve sample and primer. So one primer, this reaction, and then you must make sure that there is a priming site, okay, before you use this primer to do sequencing. Okay, with a sufficient DNA template and primer, then only you can produce a good reaction to allow you to get a good result at the end. Okay, so in this step, peptide terminator 3.1 will be used. Okay, in our lab, we are using the version 3.1 sequencing cycle sequencing kit. Okay, so after this step, production will perform according to database that we have captured all the data. Then we will go for the post sequencing cleanup. Okay, maybe you will ask me, hey Jenny, what is the difference between the template purification and the post extension product purification? Actually, this step, the purification actually, the purpose of it, the purification is to remove the excess dye. You remember that actually Sanger sequencing actually involved in ddNTP. Okay, although this ddNTP is very important for us to determine the last fragment of the sample, but this ddNTP can cause a dye block if we didn't do any cleanup prior the sequencing run. Okay, that's why this step must included to do the cleanup so that ensure that load into the machine are qualified to be analyzed later. Okay, so there are several ways to do it. We either will perform a ethanol past service or most of the service provider using. So now most of us, we are using either spin column or magnetic beads to purify the sample. Okay, so after this, the third step, the post cleanup purification, we have done. At the last step is we will load this purified sample to the analyzer sequencer. Okay. So this run batch usually will involve some high dye formamide and water, and we will look into our ABI 3730SL model genetic analyzer. We can simplify it as a sequencer. Okay, so for information in first place, ABI 3730SL is a high throughput machine. Okay, it allows us to perform more than 5 batches of run. Every run actually involved in 96 well sample. So for such a high throughput machine, it allows us to cope the international and local customer sample every day. So that's why we no need to the plate to be full, then only we will run the sample. Okay, because the volume of the sample itself is enough for us to perform many batches that we could. Okay, why this important? Because with the high throughput batches that we committed, it allows every of a customer can keep in the promised turnaround day, which is 1 to 2 days upon sample receipt. Okay, really from sent to us, maybe it's allowed another 2 more days because during the shipment through the FedEx, it needs another extra 1 or 2 days lah. Or sometimes if shipment interrupt or a shipment problem, it might go to more than. Okay, but anyhow, arrive. What first base promise is a promise that we will keep the turnaround time between 1 to 2 days for the purified sample. Okay, for purified sample. For those that unpurified sample that needs extra add-on service like purification, okay, then in that case maybe it definitely need another one day extra to perform the cleanup or gel extraction. Okay. Okay. First base. Okay. First base is quite common. Okay, quite common and quite well recognized in international, I would say, especially in Sanger sequencing, because we start up this service in 2002, like I mentioned just now. That's why we equip with a lot of experience, and we also invest a lot of high throughput machine in order for the production to eliminate like manpower handling problem. That's why we have a lot of robotic system. We have a lot of liquid handling system. This is to ensure the sample that we proceed for customer actually is in a very systematic way. Okay, so what kind of service that we usually offer? I think you all can speak it out, right? So definitely single pass sequencing is the most popular service that you all know. Okay. Other than that, we also have a single pass sequencing plus. Okay. For the plus service, actually, we allow customer to send unpurified PCR product to us, but with some extra charges. First base able to perform a purification for you. Remember, bear in mind that this single pass sequencing plus service actually only refer those sample, unpurified sample that have single band PCR product. Okay, if your PCR product contain multiple band, in that case, actually you unable, we won't include sample in this sequencing plus service, because it doesn't fall under the category of the plus service. Only the single band PCR product unpurified sample we can choose to use single pass plus service. If perhaps you say, but I really want to try this sequencing plus, but my sample have. Is there any alternative way for me to enjoy the same price with the plus service? maybe you can, okay, self-cut, okay, the gel band yourself into the 1.5 ml tube, and then you send. If your multiple band, you cut it yourself and then you send to first base. In that scenario or in that situation, yes, we still can consider customer to choose these plus services. Okay, yeah, this is what we would like to point out. Okay, if customer side, you have a very huge order, which is more than 80 samples, okay, and prepare in a plate format. In that case, you can save your money by choosing the high throughput single pass service, okay, which is the number 3 service, okay. So remember, 80 and above, this service will be entitled for you to get a better price, okay. If you want to know further, then you can always refer to our distributor, Genetica, okay. For the rest of the service, not much, not so common, but maybe I just go through. So we also offer primer walk, okay, which is some of the plasmid or insert sequence maybe customer would like to know. So in that case, primer walk will be the choice for you, okay. But please remember that we only accept plasmid sample, only purified plasmid sample only for. Okay, for direct genomic sequencing, this is referred to the sample, the genomic sample, which is have a longer length or bigger size in the charges, unless the charges is high, okay. But in order for you to sequence the genomic, actually, so make the primer really specific, then only you are recommended to use the direct genomic sequencing, okay. After that, we also have a very good rapport in handling difficult template. Sequencing basically is same like single pass, but if you know your sample is difficult template, please remember that okay, because difficult template sequencing, we actually use different protocol, which is better or more suitable. I will say more suitable to suit the difficult template. This is why First Way also can say that we are proud to be one of the service provider that can perform quite well for the difficult template sequencing. Okay, so other than that, if a customer perform their own reaction, okay, you also can choose to load sample for us to run your sample. So in this service, we only manage to load your sample into the sequencer, but we are not going to perform any reaction accordingly. Okay. So for us, we always proud to say that we in first place we high quality, and we promise the consistent long read length to our customer. So what is the promise read length? Okay, basically we promise customer we can go minimum 850 bases and up to 1,000. Okay, if the sample actually go for plus service here, okay, means that first base handle from A to Z, the purification to the of the loading sample. So we can quite confident that we can get maximum length, provided if the concentration of the DNA is sufficient. Okay, so turnaround day, as I mentioned just now, 1 to 2 working days upon receive the sample. Okay, we have a very wide range universal primer you all can check website. Okay, so the website actually, sorry. Okay. So the wide range of the primer sequence actually included in our website. So if you want to know whether your primer is entitled or in the list, you always can refer to our website. Make sure the sequence of the universal primer is totally with the one that we use. Then you can choose not to send the primer lah. You can just use our in-house universal primer. Okay, that will be totally free for the primer. I mean, using the primer, not free for the reaction. Okay, okay. Promise that we have 100% quantification for your DNA template. Okay, meaning to say that every sample that's come into the check the amount of the DNA before we proceed to the next step. Okay, so that's why some of our customers receive some email message telling that, oh, maybe your sample is low con. Okay, amount DNA is not enough. Okay, why is this happen? Okay, this is because we have our own mechanism, or we are using our fluorescent detector machine to check the DNA quality of the DNA itself. So in case the sample it didn't fall under the benchmark that we set, so what we will do is we will always refer back to our customer whether they want to proceed or not. Okay, so we want to make sure that there is a best effort guarantee for each reaction that you have paid. We don't want customer to think that you pay something which is wasting. That's why if the concentration is not fall under to guideline that we set, so what we will do, we will always refer back to you first. Okay, if allow us to proceed accordingly, then we will proceed with our special protocol. Okay, this special protocol I will calibrate more in the next 2 slides. Okay. Okay, and we are proud to telling all our customer. Okay, we have a very prompt technical support team. So in order to get your issue solved, so usually it will involve our technical support officer to helping you that to troubleshoot all the problem that you are facing if you're using first phase service. Okay, so okay, from this statement, okay, industry leader, first phase also an industry leader in sequencing that can handle difficult template, as I mentioned just now, high G or homopolymer. So this kind of difficult template, we have our own protocol to solve it. Okay, that customer must let us know in upfront if you know your sample is difficult template. Okay, so after all the sample is been acknowledged, then can be proceed. So what customer will do is, I think probably will wait for the result, right? So what we actually do is now we have our own online portal. Actually, if you all prefer that in sending the order form in soft copy, actually, yes, you are allowed to do that. But in local, actually, what we do is we are more prompt to recommend our customer to use the online portal. This is to help customer to input the data faster and to get the result archived in a very secure folder, server. Okay, so there's no right or wrong. Okay, actually just depends on your preference. Okay, if you are not using the online portal, how we still will email you the notification for you to get a password to download the result from the link. Faster, okay? Because if you receive a quantification problem or information not tally. So in that case, our customer care department, there will be a representative to send you or to keep follow up. Okay, keep follow up the information from you to make sure what actually is the information. Okay, that's why whenever you send out the sample, please make sure that you always check your email. Okay, because for current practice, what first we will do is we will always communicate through the email instead of call, because international is impossible for us to call every customer, right? So therefore, email one of the channel, which is the best channel that we will go through your site and double confirm and verify the problem with you. Okay, if there is a add-on service, okay, like quantification before extraction, purification, or cleanup, our lab will perform this add-on service first. Okay, after you perform this service, you definitely will get a report, okay, from our side. Okay, this is to inform you how is the progress after purification. If there is a needs that we need some such as we need to know which target size that you actually want us to provide, then we will communicate through the email as well. Okay. So after everything have been done, we will done DNA template quantification. Okay. Fluorescent detection from our all different measurement, we will locate the different accordingly. So in first place, we have in general you have 4 protocol. So first will be the normal. Second, we are quite we can handle the of the market. So fourth is the maximum protocol. So maximum protocol is the protocol we use to handle the protocol. It does 100% enhance of the 100% or not. Actually, it doesn't promise. For our experience, actually, for the maximum protocol, actually it works for 50% sample. Meaning to say that if your sample is low con, there will be 50% you get a good result if you are using this maximum protocol under our facility. So some of the customer, they might curious or they might have question. Before, maybe last batch, I sent a few sample, which is fall under the low concentration sample. Okay, you manage to get a good result. But in second, when you resend again, or will send other sample, also fall under the low con amount DNA sample. This time, you very poor or reaction. Result. So in that case, maybe you will question why both also low con, but it gives the impression like it's not consistent. Okay, so in this case, this is because your initial sample actually already not achieve the minimum requirement under our. That's why in this protocol, we just want to try our best to proceed or perform for our customer to do the best we could. But the fact is, a reaction is all about chemical reaction. Okay, so that's why in this case, the promise benchmark is 100%, you can get the good result. Only if you can give a sufficient amount of DNA, then the confidence level of the reaction for us to deliver the good result will be increased. So this is that I will highlight to you all how to maximize your sample to get a good result. Okay. So after we talk about the sample, the protocol, then this is the step which is quite common that I mentioned just now. So we will perform a cycle sequencing. After that, we will do a post cleanup. Okay. And lastly, we will look into the analyzer, and then we will ask our technician to provide the result analysis based on the software, base caller software. Okay, so in this process, okay, maybe customer will will try to ask whether we have any quality control benchmark. Answer is yes. Okay, so what we will do is every run batch actually we will include a control to ensure that the batch actually is working well. Okay, in case if the control is not, we will do a internal repeat for the whole batch of the result. So this is something that first base want to ensure that all the result that we release to customer actually is up to our standard. Okay, so after we rearrange everything, if the result actually fall back to the expected result that according to the technical person, so what they will do is they will release the result to our customer. Okay, here we also would like to share about how to read the DNA sequencing trace, because I think the result, okay, maybe you receive it, but you actually don't know or not sure how to analyze it. So at the end, probably you will just use the FASTA file to just blast or align it, or end up with perhaps you not even know whether this reaction actually reliable or not. Whenever you receive your sample, first thing, please try to your sequence with the software. So in ABI, actually, we are using sequence scanner. Okay, this is a free software that you can actually download from the ABI website. Okay, or in every of our result email, actually we include for customer to register yourself and to download the installer. Okay, after you download it, you just open your sequencing result under this software. Okay, what is the benefit of this software? Okay, since this is something that come out from ABI, okay, definitely they actually will design according to their machine. Okay, so whenever you send the sample to first base, okay, at the end, expect that there will be two files will be sent to you. Okay, so one is the AB1 file, will be the SEQ file. Okay, you can see from here. So trace AB1 file actually called trace file. This file you can read the electrophotogram, how's the peak and what's the size of the peak. So in this format, you can preview or you can see the sample of the size, whether they give a good result immediately. Okay, because it's performed in a graph way. Okay, so for sequence file, which is FASTA file, this is a normal file that normally customer will use for the NCBI checking. Okay, so if you're using the sequence scanner, okay, under ABI, it's a freeware. Okay, I'm not mentioned it, I mentioned again, it's a freeware, so feel free to download it. Okay, so see actually a QV bar display at the top of the electrophotogram. So plays very important role, which is it indicates the quality value of the sample or of the result. Okay, so for this particular diagram that you see here, actually is a very reliable result because the QV quality value is very high. So when it is high, it will display in a blue color. That's why a blue bar color as well. Okay, so if you see your sample is fall under the blue bar selection, okay, actually it is telling you that this result actually give a very reliable result. Okay, so QV, what is QV? Okay, QV is quality value, okay, and it's measure the confidence of the base caller. The highest QV that you get means that the more reliable for you to accept this sequencing result. Okay. So another term is called CRL. Okay, for CRL actually it represent the longest uninterrupted stretch of the high quality base call. What does this mean? Okay, so first of all, CRL usually indicate the longest QV bar that you can see from the diagram, like here. Okay, so if the 600 base pair, let's say, take this example. Okay, I can ask, if you have more than 600 base pair in the chromatogram, does it mean that it's a long and good CRL? Okay, can you say like that? Okay, actually, for your information, if your CRL is long, yes. But please put in another way, okay? If your original sample actually just up to 400, but you get a CRL wrong, actually it doesn't give a good conclusion. Why? This is because this represent that, or this giving you, maybe the sample that you are looking actually is not the sample that you provide to expect. Sample maximum go to 400 base pair, but this CRL is very good, yes, but it's not belongs to yours. So in that case, please alert or please beware that we are not just blindly say that CRL is long, then it's better. So it doesn't mean that, but definitely CRL plays a very good indication for you to understand. Okay, if the CRL is long, actually it represents that this is a high quality base call sample. Okay, so QV actually they are tracing according to the score. Okay, you can see some score here, 3 score. So CRLs also you can be found in the sequence scanner. Okay, so therefore, base calling actually is a machine that very important to read the fluorescent signal. That is where that we can understand our sample, interpret our sample precisely. Okay, so machine can read and analyze it, and then this is rely on how confident this QV bar give you. Okay, so this is the quality control that we always emphasize. We will put a control reaction in every batch, okay? And our DNA sequencing analysis software is version 5.2 with KB base caller. This is just for information if you wish to know. So it doesn't really so important for the user. Okay, this is just extra knowledge for you to know. Calling also important when you are doing the SNP hunting. Okay, because it allows you to authenticate the SNP. Okay, the software automation allow for the mechanized hunting. Okay, so therefore, so base calling playing their parts in the Sanger sequencing result very well. Okay, so let me go through this also. So okay, I would like to share also some important or some pattern of the result that you guys usually encounter. Okay, so by identifying most of the pattern of the result, you can slowly teach yourself or educate yourself how to identify. So whenever you see your chromatogram, if it is a very clean trace space, okay, with a very even space peak, okay, and without any noise background, in this trace actually is considered is a clean trace, okay. So when you get this kind of trace, when you open in your sequence scanner, you should be happy, okay. Means that this is a good result that you can expect. However, if you see something like this in your result, okay, after they return back the zip file, and then if you see something like this, okay, in your mind actually you can try to find a cause, okay, or maybe predict what actually happened. So in this case, you can see the peak actually is very low and very noise. Result supposedly is good, okay, from the beginning. Okay, suddenly there is a point which is you see a drop signal off here. Okay, what we can assume that this actually is because the big type have difficulty to pass through. So either we will use our try to enhance, see whether there's any improvement. Okay, if there's any improvement, okay, what we can do is we will tell customer maybe in the file which is under difficult template protocol. Okay, so next, if you see some of the signal actually gradually lost, like what you can see from the diagram, okay, this kind of pattern actually give you a hint also. The ratio of the primer and template maybe is not proper. It doesn't mean that they don't have a cycle reaction. It did, but just that the ratio is improper. That's why the signal is not strong enough. Okay, in that case, you will see the signal not good along the whole sequence. Okay, another possibility also might happen because of contaminant inhibit during the cycle sequencing reaction. So is quite similar or quite close like the weak signal that we saw just now. Okay, so in order for us to troubleshoot further, definitely in what we will do is we will reaction to verify. Okay, to verify, because science is based on what? Proof. Okay, what we do is we need to verify by repeating few reaction first before we let our customer understand what is actually happening. Okay, there's another pattern that you can see, which is involved in top-heavy. Top-heavy issue actually is involved like some excessive template, means too many sample is used during the sequencing reaction. Okay, maybe you say, oh, why too many sample? Okay, means to saying that the volume or the amount of DNA sample may be the sample is too much. Okay, what we will do is we will try to see whether we can get better result after we further dilute the sample. Okay, so there's another top-heavy pattern from here. So this is a saturated signal by short extension product, which is exceed the layer. That's why you can't see the peak of the sample. Okay, so in this case, what we can do is we will also try to rerun this sample again before we release to our customer. Okay, again here, this is the link that you actually can get, and then you can download from the website. Okay, or you can find from you. From there, there's a link, register it, and you can download the sequence scanner. So since it's a freeware, okay, we highly recommend customer to utilize it accordingly. Okay. That has a lot of features you can see from here: analyze raw, annotation, sequence, etc. So if you all need another section to talk about sequence scanner, actually First Wave will be happy to provide it in the next webinar if necessary. Okay, so color represent quality of value, like I mentioned. Blue color represent very good. Okay, this sequence is good to believe. Yellow color means the confidence drop to 50-50. So red color even worse, means that this nucleotide actually totally can't be trusted. Okay, means that this position, even though written as A, but maybe it's not A. Okay, so from the sequence scanner, you also can preview all the sequence in nucleotide format. Okay, so from here you can eliminate those that unrelevant yellow color or red color, and copy the middle part here, and then this particular sequence that you copy, after you eliminated the uncertain sequence, then you can use for your such as alignment or blasting. Okay, let's move on. After we know how to interpret the data and understand the cause, so now we want to let you know, okay, to get a good quality of DNA sample. Okay, please have a look on this. Okay, actually the purity, as mentioned before, is playing a very important part if you want to maximize the successful of the sequencing. So please make sure there's no contaminants, okay, inside before you send to first base, okay. So how to do that, how to make sure that you have a good quality of sample, or how to eliminate this contaminant, okay, how to make sure that, yeah, the potential of contaminants will exclude in this sample, this PCR product or plasmid. So some of the contaminant like protein, chromosome, DNA, so on and so forth that indicate here might affect the DNA sequencing badly, okay? However, okay, this contaminant actually have some tolerance amount. So if you can, this is the tolerant amounts, meaning to say that if more than this, it will start to impact sequencing result. But if less than this, okay, perhaps you still have a chance to get the the good result. Okay, so in order for you to ensure that you can get a good result, always make sure that the tolerance amount not more than this. And another one is quality of the sample is good. Otherwise, if your quality of sample is let's say your amount of DNA is very low, what we will do is we need to introduce more volume into our reaction. So in that case, more tolerance amount will introduce into that reaction as well. So that's why even though low amount DNA sample, we try to use the maximum protocol to do it, but if the quality of the sample itself have a lot of issue, or it involve other issues such as chemical reaction happen inside, it will still affect. It will still unable to give the optimum result sometime. Okay, so the best way how to make sure that you can get a good result. Okay, again back to the source. Okay, back to the first thing to do. Make sure the sample DNA is sufficient. Make sure you purify your sample. This is the most important thing to know. Okay, like if your sample is from plasmid, make sure it's from single colony. Okay, this is a correct way to do that. So if you pick some sample is more than one colony, then definitely the result will be noise. Okay, so sample plasmid DNA, definitely you will do extraction. You might use a kit method. But for our experience, we highly recommend customer to use the conventional method instead of the kit, because our conventional method will introduce a lot of human handling step. So if the person is not equipped with a good experience, maybe it will affect the preparation step. Okay, so avoid genomic DNA, assess RNA and contaminant. Okay. Elute your plasmid sample in the distilled water, and recommended not elute in TE, okay, for your plasmid DNA. Okay, this is the conventional method, okay, because as mentioned just now, if conventional, you are using conventional method, consistent might be an issue. Okay, you might see this. Okay, this is an example that we would like to show our customer. It's a good example. Okay, before that, the results show okay, but the rate is not good. Okay, it's not really up to what customer expected. So after we try to troubleshoot further, we try to use the commercial column to purify the sample. So after that, you can see the repeated result actually give a very good rate. You can see definitely more than 600 here. Okay, so in that case, showing that handling issue to avoid, actually you can prefer to use a commercial column to get rid of this kind of issue. Okay, for PCR product, yeah, same purification. You might choose clean up purification, magnetic beads, or exosub. As long as you purify your sample, make sure there's. If you have multiple band, the only way you can do is you use gel extraction purification. Okay, if you don't want to waste your time, you also can allow service space to help you to do the gel extraction. Just that you just need to select the add-on service. Okay. So this is a gel photo which is showing that before purify and after purify. There's huge difference. Okay. So only single band, okay, target band sample actually allowed to be proceed to the sequencing. So how do you check the agarose quantification? How do you check the sample? So agarose quantification choice, because from the gel read, actually you can interpret a lot of information from there. Maybe you can see. Okay, for PCR fragment that more than 400 base pair, always use 1% gel with the 1 kb DNA ladder. For those that less than 400 base pair, use 2% agarose gel. And don't forget to indicate your 100 base pair ladder. Okay, ladder is very important as a measurement. Okay, otherwise you won't know whether your sample size is correct or not. So this is an indication. So whenever you want to send the gel photo to us to verify, we always like to remind customer that please remember to put the marker. Otherwise, the gel photo doesn't make sense. Okay, you just do something as wasting your time only to just run a gel without the ladder. Okay, so load 0.5 microgram DNA ladder at first well, and another sample which is 1 microliter for each of your sample. Okay, so okay, this is a gel photo that we would like to highlight, or we would like to share with you all. Okay, by using this reference, actually more or less you can know whether your sample is suitable to be sent out or not. Okay, to run the gel photo actually can save your time. Okay, you can analyze whether your sample is qualified or not before you. So in this case, this is plasmid, right? Okay, plasmid actually, if you have a good plasmid, under the gel photo, what you can see actually is like this, which is displayed here. Okay, so it is a non-linear sample. You can see actually there is some multiple band, four upper at the target band. Okay, which is here. Okay, this is normal. Okay, if you are running the plasmid sample. Okay, but please make sure your sample has band. If you run your plasmid, you couldn't, or you see very faint band, smearing band like this. Okay, so the suggestion for you to do is don't send to us. Okay, please make sure that you re-extract the new batch of DNA. Double check again with the QC, the AQ agarose gel quantification. If passed, then only you send to us. This is to save your time and to save your money as well. Okay, you have the, which is you encounter like this. You observe a bundle of a smear of the gel photo. For this one, we can conclude that actually there's a RNA contamination. So what we can help to recover this sample actually is by using the RNA treatment. Okay, treat it. How you do that? Usually help customer to do that. Okay, as a add-on service. So in that case, you still can proceed it after you treat the sample, the plasmid sample. Okay, if comes to PCR product, okay, the good quality of PCR product actually also mentioned or on the left-hand side here. Okay, so in this gel photo, you can see a very specific and clear target band. Okay, no smearing at the bottom, no double band, no overexpose. Okay, so in this case, if you have such a gel photo image, means that this sample is to be sent out. Okay, for the rest of the sample here, which is at your right hand side, actually show all the example which is have poor purified PCR product, even though purified, but it doesn't match the benchmark of our QC. Okay, so smearing. Okay, so you can see some multiple band. Okay, so if you see something like this, please re-amplify it. Prepare a new batch of PCR product. Optimize your PCR condition, especially if you have smear product. Okay, it might happen because of nipple primer set. So make sure that you check all your PCR condition. Okay, it can be improved before you send to Sanger sequencing. For those that have multiple band, what is the way to save it? Can I know? Anybody can tell me? You might type on your question. Okay. Yeah, if you know the way to do that, actually, wait, let me, oops, sorry, give me a minute. Okay, if you see from here, okay, the multiple product actually can be rescued or recovered by using a simple, very gel cut, okay, gel extraction. So if you don't have the skill that handle it, okay, in first place, we can also provide as a extra service. Okay, so this is very useful information, I believe. Okay, this gel photo, okay, you can always refer to this website. Okay, here, look for question 8, and then you can download this gel photo and match it after you run your gel. Okay, this is as a reference for you. Okay, so this is very good for you to justify yourself before you send to first base. Okay, last, we come to the storage and handling DNA template. Okay, as you all know, actually, most of the international DNA sent to us by using FedEx Courier. So actually, don't worry about that. So far, all our international customer use this method, sent in room temp. DNA always can be safely transported in room temp and stable in the room temp for around 7 days. So doesn't matter. So however, please make sure that you store your sample at 4 degrees before you shipping. So once we receive your sample, actually, we also will store all your sample in 4 degrees or so. That's why this is to ensure that the sample actually always keep in a fresh condition. Okay. So for the shipment purpose, actually, it doesn't really, it doesn't of the sample as long as it's not more than 7 or more than 8 days. Okay. So for the plasmid DNA, use freshly extract plasmid within 4 months. Okay, not more than that. Okay. I think this has been mentioned just now. Dissolve your plasmid in H2O. Okay, better in H2O instead of TE. Bad quality of RNAs will degrade your sample. So make sure that run the gel. Okay, run the gel. If you see this RNA contamination in your sample, you can treat it. Okay, treat it. Then only you send to your service provider. So make sure that before you send for PCR product, before you send to us, same also, run the gel. Okay, make sure that the size is correct. In PCR product, size is very important. It did matter if the size of the PCR product is not matched with the leather. Okay, so I mean, you must understand actually what is your exact size. If your gel come up with something that is not what you expected, then you need to recall it whether any step actually goes wrong. Okay. Okay, and then before you send to first base, definitely the sample requirement is something that is very important. Make sure you read the guideline, okay? Read the guideline, look from our website and get the latest version. Even though in the Excel soft copy order form, actually you also can find from there the information about the requirement, what is the concentration, okay? Different PCR product actually request different concentration of the sample. But for the volume, definitely you can provide us more. You can give us more if you can, because in that case, for international customer, if we want to repeat, it will speed up the repetition. If we want troubleshooting or do, okay? Always recommend customer to send more, because happened before that some sample encounter some evaporation problem, and eventually it become lesser when arrived to our lab. So provide us more, okay? Don't be so stringy. And the primer preparation requirement, make sure that you send the 10 micromole primer concentration to us. Don't send 100 micromolar primer to us. For primer, we won't measure it. We will assume concentration they actually fall under the correct guideline. Okay. Okay. For the labeling part, actually pretty easy, but yet a lot of our customer actually it's not a. It is important. Okay, make sure that the lid and the body actually label the same information. One, make sure your body also one. Okay, otherwise, if the lab technician found the label is different, what they will do is they will on hold your sample until customer verified with us. Okay, this is because it happened because we assume, okay, end up with actually this is actually customer have different approach, want us to sample. So that's why now we are very precaution on make sure that the label actually is correct. So if you need any collection box, feel free to get from PT Genetica, okay? Yeah, so sending order step is quite strict. Tube can send us a gel photo if you have, label the lane number, name, and the volume of the DNA. Make sure you have marker, means it's the marker leather. And lastly, put into the collection card. If you have a bubble wrap, because service provider, they will just simply throw it, right? So you can see from here, okay, make sure the collection card indicate the order ID or at least the name, okay? Because we will match back your form and the card. If you don't write any information, the sample will forever fall under the fish until we clarify owner. Okay, happened before. After 1 month or so, nobody claimed the sample. We don't know how to identify it. So make sure that you put your name. Okay, let us know. Okay, this is here is the end of my presentation. I hope you get a lot of information from here. So may I know, is there any question you would like to know? You can actually raise your hand. Okay. Or you want to unmute yourself, also can. I believe for the question part, most of the question actually my team member already addressed it. So in case that if you didn't get any answer from us, okay, you can let us know, send to support, and we will try to let you know. Let me open some questions. I can answer some of it. Okay, there's one question from Gayan Pertila. Okay, can we use spectrum method for quantify to meet the concentration requirement? Okay, this is a very common. Basically, yes, but what happened actually is if we are using the nano drop, right? Actually, you just able to check the ratio of the sample. So you didn't actually can interpret more data from there. Sometimes the measurement itself also have some variation because we try to do that testing before, the experiment before. They have some variation about if compared with the fluorescent detection that we are using. That's why we highly recommend customer to use the agarose gel quantification, because I think gel agarose quantification can interpret more data. As you can see just now, I showed all the gel photo inside. From there, you can know the quality of your sample, whether it's good or not. Not only about the concentration, you also know whether your sample is degrade or not, whether your sample have primer dimer or not, and you can use a correct method, okay, to recover or to improve or enhance the sample in the future. Okay, from Dr. Kiba, will we have the slide from this presentation? Okay, sorry about that. For the slide, we won't share, but what we can do is we will channel or we can share the webinar recording, okay, by sharing in the YouTube channel. So if you need the recording, okay, you resit again, okay, maybe to load back to repeat again what you need to know. Actually, you can get the permission from us. We able to channel you to our YouTube lah. Okay. Wait, let me have a look on the question. Okay, can we identify the amplitude of 16 RA? I believe our another organizer have answered you. Yeah, more sequence means more specific lah. So for the barcoding, we will suggest to have you sequence forward and reverse. Okay, then you can have a alignment result. Okay. Let me check again. Trace can. Okay, there's one question from Ayu. For the clear trace with drop signal, okay, can we just go for further analysis, or we have to repeat the sequencing with difficult template setting? Here, what we will do is you can, if you see something, the pattern like that, you can try to refer this reaction ID to us, means to first base. Okay, what we can do is we will repeat one or two reaction, which is by using the difficult template setting. So in case that if the repetition is improved, then we will inform you further. Okay, we will inform you further. Then the rest of the sample, if all is put under the difficult template, fall under difficult template, then you might consider whether you want to repeat with the difficult protocol or not. Okay. Usually we will check with customer again. Can try to let us know which reaction ID, and then we will troubleshoot first. We will help you to check first before do a justification. Okay, because maybe there's another possibility that make the drop signal. But in case that if we think that repeat is help and then it's correct, the difficult template it did improve, then definitely showing that this is fall under difficult template. Okay, so we will definitely let you know. Okay. Okay, how to avoid primer dimer? Like we mentioned in the answer. Okay, you must optimize your PCR protocol. Okay, so for the primer dimer, if you can perform better by you protocol, definitely it will help. But if you already perform it and the step already come to the end, like you want to send to sequencing, okay, what we can do is the last step to help is we will advise customer to go for the gel purification, which is gel extraction. So in that case, possible also you can save your sample, okay. So you can try to purify the sample and then try to see whether the sequencing result actually turns into a better result. Okay, but again, the most important thing is make sure that you optimize the best the protocol, PCR protocol. If you find every time you also have a primer dimer. Okay. Okay, I think what kind of kit purification that you use? Okay, I'm not sure whether we answer you. We are using the meter. Okay, version 3.1. Okay. Okay, Mia Trial Novianti. Okay, you say you actually run a sample on the electrophoresis 700 base pair, but it should be 600 when you send to first base. What's wrong with my result? Okay, in that case, you need to have you open your result, your chromatogram, and check. It doesn't mean that if you receive the the FASTA file, the nucleotide or the number of nucleotide consider is the reliable. So most important thing is, as I mentioned in the previous, whether you actually check it, whether you really see it fall under reliable. If 700 actually, some actually is at the beginning sequence. Sequencing actually you must expect loss around 30 base pair bases at the front. Okay, if that one you conclude in your sequence, actually it doesn't really make sense. So that's why perhaps you will, after you really check it, actually the sequence maybe is just more or less 600. In case that if this also done, you double check already. Yeah, really my sequence is just I guess 700, but my sequence is 600 base pair. Okay, what I can recommend you to do is maybe you can send us the order ID or reaction ID lah. Then we can try to help you to double check about the reaction. Okay, and then we will try to explain to you what actually, because from the question here, we must check your sequence, your chromatogram, your CRL, and all the information before we can conclude it. Okay. Oh, Mr. Ahmad say, actually, what is the purification kit for the cycle sequencing? Sorry to tell you, we unable to tell you the brand, but what we using the purification actually is magnetic beads method lah, purification kit. So this is what we can. But as for the brand, actually is we couldn't disclose lah. Sorry about that. Okay. At least answer part of your question. Okay, there's one question from Norma say, after 3 months of sending the result to customer, can the customer go back to you, means first base, if they lost their result by accident place? Do you still keep the old result? If so, for how long? Okay. For 3 months, I think we still keep it, just that customer have to request it lah, have to request from us. Usually what we will, in production, they will keep the result in the server. I mean, in our own server about 3 months, okay? But some of the old result, we will actually archive it lah. So in order for us to look back the old result, actually it takes time. So you let us know what is the order ID. This is very important. So that we can trace back for you, but allow us some time, okay? Allow us some time so that we can really found the result back and then resend to you, okay? Perhaps it will need some time, okay? But not too much, okay? If let's say you say, well, I have 100 order ID, oh no lah, not so much. Say if you have more than 10 order ID, and in that case, a bit difficult for us to look one by one lah. So what we can do, we will try to help. If too much, then it will to spend more time lah to get back to our customer. Okay, does customer ask how long is needed to analyze what? I didn't see the end part. How long is needed to analyze? You mean our result? Okay, maybe you did a whole genome. Do you mean the sample? You did, maybe you can clarify your question again. Sorry, because I see how long is needed to analyze, then I see another question is whole genome. So you expect how long is needed to analyze the whole genome, is it? Okay. Please clarify with us. Oh, before that, maybe I check with Ahmad Afifuddin's question. What the quantification method you use for the DNA template measurement? Okay. I think I mentioned in the presentation just now, okay, we are using a fluorescent detecting method. Okay, so in that case that the variation actually is a bit different. This is because match back with our protocol. So this is the best method that we actually use. Okay, so sometimes if you compare with the nanodrop or the spectrophotometer that you your own lab, maybe the reading will be different. Okay, but actually we did do a video to explain about the why there is a variation happen and how actually first base do the measurement in details. Okay, maybe can share the link to you later. Okay, Miss Ahmad Afifuddin. Okay, later I will send you through. Okay. Okay.